Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Professor David H. Walker. He's in the Department of Pathology at the Carmage and Martha Walls Distinguished University Chair in Tropical Diseases. That's another designation of his. He's also Executive Director, University of Texas MB Center for Biodefense and Emerging Infectious Disease. We're going to talk about immune mechanisms of various parasites. So, David, thank you for coming.
No, good to talk to you. Always like to talk about these things.
Yeah, I mean, what got you interested in parasites? It's a lot of people, maybe it's unusual. You know?
So parasite is a very general term. It's something that lives on or in something and causes harm to it. But when vernacular use it, they usually talk about worms and protozoa. These are actually bacteria. Rickettsia, Ehrlichia, and Orientia are bacteria that can only survive by living inside the host cell. And they alternate between two different hosts. A vector, which is an arthropod like a tick, a mite, mouse, flea, and a reservoir host or an accidental host. So like Ehrlichia chafiensis lives in deer as its reservoir host, and humans are accidentally infected and it causes a disease in us. So these are, in a broad general sense, parasites, but not what most people mean when they say parasite.
Well, people call viruses parasites, but in the traditional parasite world, so there's worms, there's bacteria. What other kinds of forms do parasites take?
So when people who are parasitologists, we'll be talking about different kinds of worms: trematodes, nematodes, cestodes, and protozoa, which are single-celled organisms, but they have a nucleus. So they're really eukaryotes. And where bacteria are prokaryotes, they have no nucleus. So their nuclear material and proteins are mixed together in cytoplasm.
So you're focused on the parasites that are bacteria, actually.
That's right.
How have people tested that these bacteria can't survive outside of a human host, for instance?
Well, everybody who comes in ill with it, and they draw a blood culture and they send it to the laboratory, put it in a bottle or on agar, get negative results. Nothing grows, even if the blood is full of the organism. And you can only isolate them by putting them into a host cell. So you could use cell culture like they do for viruses, or you could use an experimental animal. And some of the earliest breakthroughs were inoculating embryonated eggs so they could grow in the cells of the embryo of the chicken egg. And that was a pretty clever thing to do back in the day when we didn't have biosafety hoods and didn't have antibiotics. And so that the things that enable cell culture to be done effectively couldn't be done back when they dreamed up the idea of inoculating the material into an embryonated egg.
Has anyone used light microscopy to put a cell and a bacteria in culture and watch it infect and see the mechanism of infection?
So it's been done by electron microscopy. I've done that by electron microscopy. You just take pictures and see the different stages of attachment, invasion, and some of these actually induce the host cell to do things that it wants the host cell to do, like take it in and then escape from the phagosome. Rickettsia and Orientia escape from the phagosome. The Ehrlichia and Anaplasma, they stay inside the endosome, modify it to where they won't get killed in there by lysosomes, and do lots of other things to manipulate the host defense to suppress the host cell's ability to get rid of it and to preserve this place as a place for them to live through their life cycle.
So what do you think happens in culture in the absence of a host cell? I mean, it sounds like the bacteria is mobile. It moves around and positions itself near the cell, and then, like you said, induces maybe endocytosis or phagocytosis. But
none of these bacteria are motile outside of the cell.
Really?
Some of them have means of making the host cell move them around. For example, Rickettsia, the spotted fever group, can stimulate the host cell to polymerize filamentous actin at one pole. It will move it from one side of the cell to the other, enable it to enter the adjacent cell, so it can spread from cell to cell to cell.
So the reason why I ask about light microscopy versus electron microscopy is, perhaps you could watch what happens while the cell and the bacteria are alive, where they're introduced in proximity to each other,
That's been done. There were some pictures taken by the Russians where they really did time-lapse photography and showed pictures of these cells, of the bacteria motoring around inside the cell. The steps of sticking to the surface of the host cell, adhesion, stimulation of entry, there are lots of ways to study that. And light microscopy, which is my favorite too. I'm a practicing MD pathologist, so looking in the microscope is something I love to do. The first person who discovered Rickettsia was a pathologist from Chicago named Howard Ricketts, after whom they named this genus of bacteria, which actually killed him. He died at a young age, being infected in the laboratory. So he isolated the first one by inoculating guinea pigs, so the cells in a guinea pig could maintain it. And he was a microscopist and looked very carefully, and he saw them. He drew pictures of them in his lab notebooks. It was a long time before people really figured out what they were. He would pass them through filters that would retain bacteria and they wouldn't go through. So he knew that they had to be either a bacterium or a protozoan. But at that time, the concept of a virus didn't exist. Somebody said virus, they just meant some sort of living thing that could replicate itself. Yeah, that would have included bacteria. Lots of things fit under the category of the word virus back at the turn of the 20th century when Ricketts was doing this work out in the Bitterroot Valley of Montana. So it's a long story. Some of it has to do with really understanding where words come from and where concepts come from.
So I guess through just fluid dynamics, the Rickettsia, like you said, is non-motile. It'll pass by a cell in close enough proximity where it will bind, or what's the initial contact with the cell look like?
Sure, it's got some ligands that react specifically with certain host receptors, and a lot of these are being identified. And they actually have, usually one of these bacteria will have more than one of its protein ligands, and there will be more than one receptor of the cell. It's got backup. Even though these things have tiny genomes, they've undergone reductive evolution to where their genome is like a quarter of some other bacteria. So they actually use fewer genes and manipulate the host cell to do what they need to do, and yet maintain backup systems. So if one receptor is not available, maybe there are a couple others that will. And once they get inside, they polymerize the actin to move around. It depends on which ones you're talking about. And they're the Orientia anaplasma, Ehrlichia anaplasma, Orientia, and Rickettsia all belong in the same order, Rickettsiales. But they've really got major differences among them, such as whether they have lipopolysaccharide in the cell wall or not, what their proteins are. It's a complex group of bacteria that are very difficult to study because you can't study them just on their own unless you're going to just do pure biochemistry, because they're not alive and not growing by replicating unless they're inside a host cell.
Oh, so the thinking is that they're just simply not alive unless they're in a host cell, or are they?
Yeah, they're going to die. They will die. They got to get into a host cell.
How long is it estimated that they can live without a host cell?
Well, people have done some experiments trying to figure out how they can keep them alive long enough to preserve them, to use them for research. And different organisms have different degrees of fragility, and there's decaying of their viability that begins immediately. But there'll be some live still if maintained at a very cool temperature 24 hours later. They won't all be dead, but they will not. They're really all going to die. It's just a matter of how long it takes them to die.
What form are they inside of? These come from a tick, this family of bacteria?
So the spotted fever Rickettsia do. So there are two main pathogenic groups of Rickettsia: Typhus group and spotted fever group. And so the Typhus group, which causes typhus, was probably had more effect than any other disease on European history between 1500 and 1900. Whichever army got the louse transmitted typhus first lost the war. I mean, it was really not a matter of how good the generals were. It's how when they ran into typhus, and of course nobody even knew back then what a rickettsia was, nor that the louse was a vector. So there's some insects that transmit the typhus group: fleas for Rickettsia typhi, murine typhus, and lice for epidemic typhus. In the spotted fever group, they're mostly transmitted by ticks. And Ehrlichia, they're transmitted by ticks. And anaplasma are transmitted by ticks. But Orientia, this is probably the most important of these disease genera now. Orientia tsutsugumushi causes scrub typhus, and it's transmitted by chiggers, larval mites. So you've got to be, we need entomologists to help us understand these, because while I'm a pathologist and study them carefully in experimental animals, and I've studied autopsies and biopsies of people, most of their life some of them is spent out there in that arthropod, as you said, like the tick.
Well, so step by step, what does it look like? If I'm in a tick, am I inside the tick cells? And then at the point where there's the blood meal, do they come out of the cells and then enter into the human and then seek to re-enter a new cell? Like
exactly. So they're secreted in the saliva of the tick. They're in the salivary glands, and they're secreted in the saliva of the tick, which goes into the dermis of the skin of the person. And then they're taken up in the skin of the person by phagocytic cells, like macrophages and dendritic cells. They enter the lymphatic vessels, and they spread via the lymphatic vessels to the draining lymph node, and from there into the bloodstream. And then they go all over the body in the bloodstream, and the cells they infect are mostly the endothelial cells, which are the cells that line the blood vessels all over the body of all sizes: arteries, arterioles, capillaries, venules, veins. But most of the circulation of the body is microcirculation: capillaries, venules, and arterioles. And they grow inside the cells lining these, the endothelium. That's what the organisms that cause Rocky Mountain spotted fever, for example, do.
Well, I do have one more question here, and then we'll move on to, once it's in the human host. So at the point, these bacteria are literally inside the cells of a tick, for instance, or a louse. And then at the point where you said they're in the salivary gland, are they inside cells and they come out of the cells and then they go into the saliva in a free form? Or are salivary cells going into, let's say the person, and the bacteria is inside them, and then once inside the person, does it burst out of those cells and then travel and then go into our cells? Like, at what point is the bacteria not inside a cell, and for how long, and when does that happen?
Some electron micrograph pictures suggest that they come out of the salivary cells into the saliva free, and then go in the tick saliva into the little blood pool that the tick is sucking blood out of, and are taken up by the hosts, macrophages and dendritic cells. But you have to realize it's not; these are not all the same. I'm telling you, for one, I would fit the spotted fever rickettsia.
I know.
If you like this podcast, Please click the link in the description to subscribe and review us on iTunes.
And it's the you mentioned the louse. Actually, the louse sheds it in its feces, and it gets scratched into the host, and it would be an extracellular organism at that point.
Yeah, it's just interesting because I'm wondering what kind of signaling occurs to tell the bacteria, all right, exit the current cell you're in, and then look for a new host, a new cell to go into.
Yeah, right. I mean, that's. I think we might be giving them more maybe anthropomorphizing what's going on there, because these things, they're not smart. They're just highly evolved to be able to do these things. And so I'm not sure what. I'm not sure that I'm sure there are signals. For example, if a tick is out in nature for months or years at a time, it's at ambient temperature. But when it gets onto a host, it gets the skin temperature of the host becomes the temperature of the tick. And as the tick begins to imbibe blood, this blood nutrients may have some messages in them, so that the rickettsia change. And in fact, if you grind up a tick from nature and inoculate a guinea pig doesn't get sick. But if you take a tick that's been feeding on a guinea pig for a while, like a couple of days, and grind it up and inject a guinea pig, if it's a virulent rickettsial, it'll kill the guinea pig. So there's some type of reactivation that occurs, and part of it's the temperature, because you can just take that tick from nature and stick it into an incubator at 37 degrees centigrade, leave it there for a couple of days, and they will have acquired some of the virulence. Not as much as if it had taken a blood meal, but enough to cause disease.
So what part of the of the bacteria's life cycle are you focused on? What are you trying to figure out in particular?
So I've been working with Rickettsia for 47 years, and I've done a lot of different things with them. At this point, I'm 77 years old and am focusing down on trying to accomplish something practical in life. And there are many scientists who are out there studying signal transduction, how these Rickettsia with small proteins can use the same protein to do many different things. They're multifunctional. They call them moonlighting proteins. But there are people that are doing incredibly sophisticated work. My personal interest at this point is vaccine and diagnostics development. Really, having a practical impact on some of these diseases. And people have been trying to do that for decades. One I'm working on very hard is scrub typhus caused by Rickettsia, and it affected 18,000 troops in Asia and the islands of the Pacific in World War II. It was a major cause of febrile undifferentiated illness in the Vietnam War. And people have tried to develop vaccines for 8 decades and don't have one, even a candidate. And that's one of my goals, is to actually make the steps that are going to make a vaccine happen for scrub typhus. The other thing that I'm working very focused on doing, and I'm working with a very good colleague, Dr. Jerry McBride, and he's the genius behind that one, on vaccines for ehrlichiosis. There are several ehrlichioses. And the diagnostics, we've got some really good ideas and some good preliminary data, because the problem is these diseases are diagnosed by detection of antibodies the person makes against the organism. And the antibodies don't appear until too late to be useful, really. Say for Rocky Mountain spotted fever, you need to treat the patient in the first 5 days of onset of illness. And they don't make antibodies until day 7 or later. And so we really need new tests that will identify and make the diagnosis at the time when you need to make the decision of what therapeutic drug to give. And people with fever, they always get antibiotics. But actually, there's only one that works against Ehrlichia, and that's Ehrlichia and Rickettsia, and that's doxycycline. There is some evidence that there may be another antibiotic that you could use against Ehrlichia, but the ones that are actually used that work are doxycycline. And patients have got sepsis, which is a febrile illness with lots of severe clinical manifestations. They get treated with all sorts of antibiotics that don't do anything to Rickettsia.
So what's in terms of a vaccine? I know it depends on the bacteria. Would you try to stop initial entry into our cells? Would you try to stop once they're inside of our cells, proliferation, or would an immunotherapy work? I mean, what's been the thinking, and where does science seem to be making headway to treat some of these bacterial diseases?
So for Rickettsia, it's the outer membrane proteins that are used in entry to the host cell. And we call them outer membrane proteins A and B, which Typhus group does not have outer membrane protein A. The gene names that encode those proteins have got very different. Surface cell antigen 0, 1, 2, 3, 4, 5. So that's the main idea that people have had for Rickettsia. As far as a subunit vaccine where you just use one protein. The idea of a live attenuated vaccine just means attenuating it, finding some gene that the organism has to have to make you sick or to grow fast enough to make you sick, and eliminating it. And these organisms have been very difficult to manipulate genetically. If you can imagine, you go and knock out a gene, and you try to get this organism to go into a cell and grow, and you've knocked out the gene that it uses to get into the cell. You're not going to have. It's not going to be easy to accomplish. For scrub typhus, it's fascinating. People have looked at all sorts of proteins, and the major surface protein is a 56 kilodalton protein, and it's plays a role in adhesion to the cell and entry, and antibodies to it prevent the illness. So you can immunize a mouse with antibodies to the P56 of a certain strain of Rickettsia, and that mouse will be protected. The problem is there are many, many different strains of Orientia, and they have different 56 kilodalton proteins. And so if you're immune to one, you're only immune to one. And the other ones you're exposed to, you will get no protection at all. So how to handle this? And the United States Navy worked on this for a long time because of the Vietnam War and the Marines out there and the legacy of the severe effects it had in World War II. And they didn't succeed in getting a vaccine. Right now, there's the work that's going on that looks as if they're making some headway is in Korea. And they've taken these same cell surface antigens, so-called SCAs, and they found there are several of them, and you have to find one that's conserved among the different strains so that vaccination will really work. And they found one called SCA-A, which has some promise. And they've also tried to take the conserved part of the 56 kilodalton protein and make a vaccine out of it, and they've had some success. My idea is something very different from that, and I'm going to try to get the money to do it. And got an application that went in. It's being judged this week. It might get me started, and on the other hand, it might not get funded.
How much variation is there in strains of bacteria for a given infection? Like, has someone been, have you looked at the cells of someone that's been infected, and have we observed many different types of Rickettsia, for instance, or Ehrlichia, or I know again it depends on what's infected.
It depends on where you want to draw the line between one species and another. That's an area of contention in which I have one point of view and some other people have other points of view. We have lots of names of Rickettsia, and they are more closely related to one another than bacteria that have the same name of a single species in other genera. But we have many different variations, particularly in spotted fever group, Rickettsia, whether you call them different species or different strains. And they are transmitted by different ticks and have different distributions in the world and cause different severity of disease. But for a given strain, they don't vary. They just stay the same. So, it's not going to change. In fact, that's true of the Aurantia In fact, there's so many different ones out there that this particular strain does not change.
If I'm bit,
let me tell you the difference between that. Anaplasma has a mechanism by which it just changes its surface all the time. It just, by the time the immune response recognizes one set of genes it's got on its surface, it changes them to another one. So they've got different strategies for survival.
So if I'm bitten by a tick and I get spotted fever or one of these other conditions, is it going to be a big variation of the bacteria that are attacking my cells? Or I know, like you said, some of them are very good at evading my immune system, but is there going to be on a genetic level a lot of variation in the bacteria that have been
the one you encounter is not going to change. It's going to stay the same. But there are others that you could encounter that are different.
Well, I guess for a given infection, that's good because it's a little bit less hard to combat it. If I was infected by a bacteria that had many different strains and variations, it would probably be really difficult to get rid of it, because if I preferentially kill one, the other strains would rise to prominence and keep infecting.
Yeah, but you start with one strain, and either it changes or it doesn't. And the Rickettsia don't, but the Anaplasma do. I mean, there are plenty of examples of bacteria that persist by changing their antigens. But there are others that don't do that at all. And Rickettsia and Ehrlichia and Aurantia do not.
Okay, got it. Well, very good, David. We're just about out of time. What's the best way for people to find out more about your work?
Well, I published about 360 articles. Go out and read them.
Nice.
It's they're book chapters and reviews, and they're all out there in the public publications.
Okay. Well, very good. Thanks for coming on the podcast. I really, really appreciate it very much. It's very interesting stuff.
You're more than welcome.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.